All right, here we are for another AP Biology video. This one is going to be about randomness in evolution, in particular, something called genetic drift. But let's start with this randomness in evolution thing first. You just recently watched a video about these rock pocket mice, and this guy had something interesting to say about randomness in evolution, right? He says that mutations occur at random through our DNA. Every new organism is born with a new set of mutations. But while mutation is random, natural selection is not. And this is an important idea. And as he says in the video, it's a common misconception, right? So let's start with the randomness part. Of course, mutations happen every time that you are going to make your gametes. DNA polymerase, which copies the DNA, right, during the S phase during meiosis, it makes mistakes. And a lot of those mistakes get fixed, but some of them slip through. And so every organism ends up with 10 or so mutations that are brand new. And these are important for evolution, right? Because this is one of the things that ensures the variation in all the individuals in the species, right? And so in this particular example, there was about the rock pocket mice and how they have their different coat colors. And they went and they found the mutation that makes the dark mice have dark fur, right? It's in the MC1R gene, and it produces an overproduction of melanin, basically, and it makes a dark colored mouse. Now, that's random. And one of the important things about the randomness here is that those mutations are happening all the time. So even in the light population, there are dark mice being born randomly all over the place. Not super often, but when you have a big enough population, they will pop up here and there, right? Because the light mice have this same mutation or a similar mutation happen to it. And so that idea right there about the similar mutation is the part about this that isn't random, right? So the mutations happen at random, but it is the phenotype that the mutation produces, which is the most important thing. And one of the things that they showed in the video was that, say, this mouse over here and this mouse over here don't actually have this same mutation. Maybe one of them has this mutation in the MC1R gene, maybe this one right there, but the other mouse has a mutation here, right? So the mutations that the two populations had were not exactly the same. But because the mutation produced the same dark phenotype, then it was selected for. Natural selection doesn't get to see the mutation. It only sees the phenotype, right? And so natural selection is not random. It is the survival of the fittest. It is the dark mice getting picked off more often in the light rocks because dark things show up better on the light rocks, right? And so natural selection goes by rules, if you want to call them that. But mutations, they are the random part, right? You can't predict when it's going to happen. But you also can't stop them from happening, and eventually mutations will happen all over the place, right? And so randomness from random mutations is important, the raw materials of evolution, but natural selection isn't random. Natural selection leads you to a path that is best for the environment. Most of the time, natural selection is the main driver of evolution, but there are things that are random that happen, and this is called genetic drift. Chance events that change the frequency of traits in a population, all right? And so we just did this lab where we're doing at the frequency of traits or the frequency of alleles, right? The percentage of each different phenotype or each allele in the population. And these things can change because of just randomness, right? And so these things are not technically natural selection. It is not survival of the fittest, because whatever these events are, they don't really have to do with your ability to survive. So for example, there's something called the founder effect that we'll talk about in just a minute, where one small group breaks off of the main group and starts a new colony like the red birds here, and you end up with a bunch of red birds on an island that seems like that should be natural selection, but it was just because red ones happen to show up. There's another one called the bottleneck, where some factor, but usually some sort of natural disaster that you can't plan for or really be prepared for, reduces the population, and the ones that live are random. And so that might change the allele frequencies or the phenotype frequencies in the population. But first, we want to talk about one situation from the lab, which is the small population size. And this is the most common way of thinking of genetic drift on just an everyday basis of thinking about evolution. And so in that case, let's look at this, the simulation, right? You have your kois, and they have three different phenotypes: the all red, which is the big R, the spotted, which is big R, little r, and then the white ones, which are little r, little r. And you started out with all of these settings the way they were. Most importantly, the relative fitness down here, these are all ones, so everybody lives 100% of the time, except for old age, basically. Mutations are 0, migrations are 0. The initial population was 0.5 big R's, which means there's also 0.5 little r's, right? So 50-50. You really weren't going to change these things at all. Eventually, you changed the size of the population, right? Now I'm going to skip case 1 and come back to that in a minute. In case 2, you took the little r, the homozygous recessive, and you put it all the way to 0, and then you watched what happens. I already have it on 16 times speed, and I basically just watch the red line, right? The red line, if it goes up, that's more big R's. If it goes down, that's more little r's. And so just want to check that I did it right. Yeah, there we go. I had it the right way, because it's not dropping as much as I thought it, or it's not raising as much as I thought, right? At first, it takes a while. And this is the randomness part, right? Sometimes, even though we made the little r's be dying as much as possible, 0 fitness, they are still kind of hanging out there because of the heterozygous individuals, right? But over time, clearly, we're going towards the big R side, right? This number up here is getting bigger and bigger, right? So there's our evolution against a certain phenotype, or in this case, it's genotype, but it would be the white fish, right? And clearly, natural selection will take away those fish that don't survive, don't blend in. I made them not blend in, right? Nothing actually changed about the environment. But then we'll go back and we'll leave the little r's at 0, but then we'll also make the big R's at 50. So if you're a big R, you don't die all the time, but you die frequently, much more frequently than the heterozygous individuals. And so we'll reset this and then go back to the data and push play. And here we have the same, watching the allele proportions, the allele frequencies over here, and they're fluctuating just like they did before. There's definitely a lot of randomness that, again, I'll talk about in just a minute here. But one thing that isn't happening is that gradual and relatively steep incline towards the big R, right? Like it's going up a little bit, but not nearly as much as it was going up before. And it seems to be leveling off right here. And if I give it enough time, maybe it'll even start to come back down, right? Because you still have a lot of heterozygous individuals, right? Like this is helpful. Now, a lot of this is randomness, right? Like as I'm talking, I'm watching it, and for a minute there, the big R started to be bigger than the big R little r. And like, how can that be? Well, it just happens to do with coin flips, right? Like if you are a big R in this situation, you live 50% of the time, and 50% is a coin flip. And sometimes the coin flips go heads a bunch of times in a row, right? And so overall, the longer you make this go, the more closer to the center it's going to stay. It's still skewed towards the big R's because big R's still live sometimes. But the heterozygous, it keeps the little r's around, right? And it makes the diversity more even, right? And so those are two of the cases in the lab. But the last case was you were supposed to make a bunch of small populations and do it a few different times, right? So you were supposed to go down here and make it, I think I said 50, but maybe I made it 25, but I think I said 50. It was supposed to be smaller, right? And then you would go through and then you would run it again and you would watch what happened. But this particular simulation, it doesn't show what I wanted to show as well as it could have. And in fact, the biggest flaw with this thing is that the size of the population that you start with actually isn't all that big. Maybe 200 fish in a small pond is big for a fish pond, but it's not big of not a very big population for like real life. And so 50 is small, but it's not that much smaller than 200, honestly. But one of the things that you were supposed to see is that there was a lot more randomness. And again, this one doesn't work that well. So I'm going to switch over to this simulation, which is, it's kind of the same thing. It's actually a lot of the same things, but it's just a little bit different layout. And I like the fish one because it makes you go slower, actually. You don't want to just rush through everything all the time. It's kind of nice to watch the red line go up and down. But right now, I don't want to do that. So I'm going to put this at 1 population. And here we have n, which is the size of the population. It's starting with 100. The frequency is, again, 0.5 for the big A. The big A sub 1, which is the big A. And then they have the sub 2. That's the little a. But again, we're starting 50-50. Big A's, little A's, big R's, little R's, whatever. The fitness is all 1, all the same. Mutations are 0. Inbreeding is 0. Bottlenecks, we're not doing that. And there's no migration. No migration right there, right? So this is all the Hardy-Weinberg simulation or the factors that Hardy-Weinberg says has to be there. And of course, Hardy-Weinberg said if all these things are true, then the allele frequencies should stay the same. But if I hit go here, you get a line. I forget what the blue line was. The heterozygous individuals, I believe. But what we want to look at here is the yellow line, or the green line up here. That's the frequency of the little a. And then down here is the frequency of the big A, the orange line. And you'll see that it only took like 30 some odd generations for the little a to take over the population completely. And the big A is gone completely. But if I hit go again, I get a totally different answer now. And it still made the, and this time the big A became 100%. The little a went completely extinct. And it took 100 and something generations, right? And if I keep doing this, look at the differences in the up and down, the fluctuations of the alleles, right? It's all over the place. This one especially right here, right? It goes up almost to 100%, but then it drops way down to almost 0, and then it goes back to 50-50, and then eventually goes down to 0, right? Like the fluctuations, the swings. This is all because of coin flips, right? The computer is just doing tons of Punnett squares. And every time there's a choice, a big A and a little a, it does a coin flip. And sometimes it's the big A and sometimes it's a little a. And so as we know, smaller sample sizes give you more randomness. So let me now change this to 1,000 and hit go. And the swings are not so much. It's still, here's one that the little a is close to 100 and the big A is way down here. But still nothing went to 100%. Or 0%, right? It's, oh, that one did. I went a little too fast, right? So there is randomness still. But let's say we make the population 10,000. And now, look at those. It takes my computer actually a little while to generate it after I click. It's a little slower. But way less randomness. So on the lab, a lot of people in their very first, in case 1, where you were doing only 200 fish, people still got some, you were supposed to get it to not evolve, right? Because Hardy-Weinberg says that if you were to have a big population, which theoretically 200 would be a big population, and you have everybody be equal, and everybody be equal in terms of the relative fitness, and no mutations, and no, and we have random mating, assortative, I can't even say that, assortative mating is 0. That means that everything's random, right? Hardy-Weinberg says if these things are all the same, then the allele frequency should stay pretty consistent, that you should not get evolution. But people were getting evolution. They were getting it because 200 isn't a very big population size, right? And so if you could have made it to be 10,000, then you would have seen that the allele frequencies stay right near 50-50 like the whole time. And when it gets crazy, it's only going up to 0.53 or down to 0.47, right? A big population size really makes the genetic drift go away. So the smaller the population size that you have, the more likely that just random coin flips during meiosis and during sexual reproduction, right? We don't know which sperm with which egg, that those are the things that are going to actually make it look like evolution happened. But it wasn't natural selection, right? And that's the key, that when the big A took over for the little a in the simulation, that was just because of coin flips and because you had a small population size. That in a big population size, those things should not happen, right? And so this is the basic idea of genetic drift. Random events, as simple as coin flips during meiosis, can influence the traits of a population and make it look like, well, and it does make evolution happen. but it's not natural selection, right? And notice I'm saying evolution is different from natural selection because they're not exactly the same things. Natural selection drives evolution, but evolution is just the change, and there's lots of ways to change. So let's look really quick here at some more examples of the founder effect and bottleneck. Again, the founder effect is when a small population breaks off to make a new group. They found a new village or something like that. And for whatever reason, just by chance, the individuals who picked up and moved, they happen to have a bunch of different traits, right? And so in this case, the red bird happened to get maybe blown over to this island, and you have this whole island full of red birds, and you would come back and you would say, oh man, there must be something really good about being a red bird on this island and bad about being a yellow bird because all the yellow birds are gone. But that's not natural selection. That's just because the red birds happen to be the one that colonized that area. And so this makes the gene pool change, and it makes a small population. And we just talked about how a small population is going to affect could affect the randomness inside the group. And so there are examples of this from humans that are pretty interesting. There is a genetic disease called Ellis Van Creveld syndrome. I don't know if I'm saying that one right, but it's a type of dwarfism, and it makes you have really short arms and lower legs, like your shins are super small, I guess. But it also makes extra fingers almost all the time, right? And so this is it's a recessive genetic disease, autosomal, not sex-linked. And in these population of Amish people, right? And Amish people are interesting because they kind of isolate themselves, right? They've been kind of trying to live the traditional lifestyle before they had modern things like cars. And so they keep to themselves a lot, and they marry within their own group. Amish people don't leave the Amish country too much. And in any case, there is a really high incidence of Ellis Van Creveld syndrome in the Amish population, especially there's little groups of different Amish people, and there's this one group that is like the most old school, and there is a very high amount of this going around. And it turns out that this is the founder effect, right? At first you would say, well, what is it about living over here that makes everybody have these mutations? But it just so happens that there was 200 original founders, and there was one gentleman. They literally, because the Amish actually keep a lot of really detailed family records, like genealogies and things, they were able to find literally the exact person who had the recessive trait. He was a carrier. He didn't have it, but he was a carrier, and I believe he had a lot of children. And then so it kind of spread, right? And so the more children he had, the more other kids in the family were carriers, and then they went off and had their own families and maybe made more carriers. And then the more carriers you get, the more likely you get two carriers together. And now you have Ellis Van Creveld syndrome. I couldn't find the exact numbers, but in the normal population, it's like a 1 in 100,000 chance or something like that. 1 out of 100,000 babies is born with this mutation. And I couldn't find a similar number for the Amish, but I was able to find that this particular Amish population has 12% of the population are heterozygous, right? So 12% of the people are carriers for this disease, which has got to be way, way higher than the amount of carriers out in the regular population, right? If 1 out of every 100,000 babies gets 2 little A's, there can't be that many carriers out there. But if you're from this one population and you are founded by a group that already had a high percentage of this mutation, then it leads to a high percentage in your population. This is the founder effect. This is actually, the founder effect is evident in humans in an even bigger scale. This map shows the likely migration paths of humans out of Africa and some of our ancestors. The yellow here is Homo erectus, and they were nicknamed the Great Walkers because they were really the first completely upright of our ancestors. They are basically hairier humans. There are some other differences, but the yellow, they spread all over Africa, of course, and then into the Middle East. And then eventually they spread all the way over into China. And they did actually come all the way over here into Southern Europe. Not too much. They didn't like the cold a whole lot, so they didn't go that far up. But they were all the way over here as early as, or as early as 1.8 million years ago. 1.8 million years ago, there are Homo erectus ancestors all over this area of the world. Then somewhere around this area, a new species arises called Homo heidelbergensis. You don't really need to know that other than these ones walk out and go into Europe, and they are better with the cold, and they turn into Neanderthals. So that's this one right here, Homo neanderthalensis. The Neanderthals are in this area, right? And so about, 600,000 years ago, I think is where it started. But about 100,000 years ago, a group from this area right here starts to walk out of Africa and up into the Middle East. And that's what the red lines are, right? Before that, they spread all over Africa. So they're showing the yellow here, but they have the arrows showing like, oh yeah, they eventually cover up everything. And as they go, they actually do encounter some Homo erectus individuals, especially down in Africa. And then they do encounter Neanderthals in what is basically the Middle East and over in here, right? As far as we can tell, the modern humans, Homo sapiens, they just straight up outcompeted the Homo erectus. They were smarter and were better with tools, and so they were just able to survive better. But the Neanderthals were actually pretty smart for, they're not the cavemen that people kind of thought they were a long time ago. And so some Homo sapiens actually mated with Neanderthals, which is where the Neanderthals actually weren't really outcompeted. They were partially outcompeted by the smarter modern humans, but also the populations mixed together, that they were bred out of existence basically and kind of merged all into the humans. Now, what does this all have to do with genetic drift and the founder effect? The group that makes it out, basically these groups right here, right? This was a founder group that set out from Africa about 100,000 years ago, like it says right here, right? And so the rest of the population, all of this entire part of the world, has a much lower amount of genetic diversity than people in Africa. Native populations from Africa have the most genetic diversity out of all the human groups. And one really interesting thing is that if you take away the identification of the DNA samples, and you have DNA samples from people all over the world, and you were just to take these DNA samples and group them together by like, all right, these guys all have similar DNA, these have similar DNA, you would get 1, 2, 3, 4, 5, 6, 7, 8 groups. 8 groups of like founder groups, basically, right? These 7 are all from Africa because Africa is friggin' huge. And so as the original modern humans that started around here somewhere, they spread out. A founder group sets up here, a founder group sets up here, a founder group sets up down here, all over the place. Those groups kind of, because Africa's big, they don't interbreed that much, and so they end up having DNA that's similar. 7 of these groups are all from Africa, and that means the rest of the world is that group, right? The amount of variation in the rest of the world is extremely low compared to that of Africa. And that's because of the founder group, right? The founder group that went out to found the whole thing was much less diverse than that of the entire continent that they came from. We have more evidence to show that there's effects of these migrations, in particular over here into the New World. The New World was the last to be colonized, and it would make sense that an even maybe smaller group set out from somewhere in here and eventually goes to over into the Americas. As I'm saying that, I can't help myself from pointing out that there are other options to this, that it didn't have to be this. There's also evidence that some people were from here and they hopped along the islands chains and along the coast, and that they did that actually maybe even earlier than the people who walked across the ice bridge that you probably remember being taught about before. But in any case, the point is that small groups eventually made it over to the New World. So this map right here shows the distribution of blood types in native populations, all right? It's important that it has to be native populations because we don't want to have migration. We want to just see the original group. And this is for the blood type O. And right here is the blue. And basically in native South American and Central American populations, almost everybody has type O blood, right? And so you would say to yourself, man, what is it about living in South America and evolving that makes type O blood be really good? But the blood types don't really mean anything. just recently with COVID, there's a slight indication that some blood types are more susceptible to severe COVID. That's the first time that I've ever heard of blood types actually meaning anything. Most of the time, blood types don't mean any, they don't give you any survival advantage. So why would everybody in South America have all the same blood type? Well, they were settled by a group that had an abnormally high amount of type O blood types. We don't know why they had a bunch of type O blood types. Why did everybody in that first group have similar blood? We're not sure, but it didn't reflect all the other blood types, because over here, right, the native populations over here, they're only 50% type O or 60% type O, right? Like there's been a change in allele frequencies, and it wasn't natural selection. It was a founder effect. Similarly, this is for type B blood, and in the New World and in Australia, you have a basically 0 type B blood. Meanwhile, over there in Central Asia, there's a much higher percentage of type B blood. Again, maybe you would think, well, there must be something bad about having type B in the New World and good about type B in Central Europe, or not Europe, Central Asia. Did I say Central Europe the first time? I think I did. Central Asia. It's not natural selection, though. It's the founder effect. It's genetic drift. It's just a random group settled there and they spread out, and now the population reflects that initial founding population. So the bottleneck effect. Bottleneck is when usually a disaster, something that you can't plan for. Like I said, it usually has nothing to do with your fitness, right? And so if you are, a bunch of, mice, I don't know why I go to mice, and you're just living over there on the coast somewhere, and you're having a great time, and natural selection has allowed you to adapt to be great to living in your environment, and then a tsunami comes by and wipes out 70% of your population, right? Just washed away. That's not because of your genes. That's not because of your ability to survive on a day-to-day basis. That's just dumb, bad luck, right? But it happens. Right? Tsunamis happen, earthquakes happen, fires, droughts, all kinds of different natural phenomenon that are random, right? And so in the picture, you have the blue and yellow and red population, and then the bottleneck happens and only a bunch of blue and a few yellows. And so you end up with mostly blue and a couple yellows in the final population. It makes it look like blue is awesome and red is bad, but really it's just luck that the blue ones happen to make it out, right? Before the red and the yellow ones made it out of there. A great example of this are cheetahs. All cheetahs are basically identical twins. There is almost no variation between them because the Ice Age was really rough on them. And then humans have been hunting them. And so their population has gotten so small that really they have no choice but to mate with the ones that are around them. And the ones around them are already pretty genetically similar. Now, this is an interesting point to talk about for inbreeding, in that inbreeding is definitely associated with birth defects, but it's not because it makes birth defects. It just means that if you happen to mate with one of your relatives, if you're a carrier, they also might be a carrier. And so the chances of having kids that have weird diseases goes up. And then, of course, if the cycle continues, then you just amplify those same mutations, keep getting reinforced and reinforced and reinforced. But in humans, there's no other natural selection other than being able to just be born, right? A lot of those mutations that might have been bad from inbreeding, they wouldn't necessarily make you not survive. So the mutations are allowed to pile up. But in the cheetah population, only the ones with the best genes have survived. So they're not passing bad genes to each other. There aren't any weird mutations being amplified, because every time a bad mutation happens, it gets weeded out immediately, right? Like natural selection takes care of that one. And so you're getting the strongest cheetahs mating with the strongest cheetahs still, but there isn't any variation. Therefore, there isn't any way for them to respond to changes in their environment. Their environment hasn't really changed too much naturally, but then, of course, humans have been changing their environment artificially. And so this leads to the cheetahs being especially endangered because they don't even have any possibility of someone out there or some cheetah out there having some cool mutation that would would rescue them from whatever environmental doom that natural selection and humans have kind of laid out for them, right? So the bottleneck is another example of randomness that happens in natural selection. The founder effect happens, and then just good old randomness from meiosis and mutations, and this can influence the traits of a population and lead to evolution that isn't natural selection.